Good evening, good afternoon, good morning. A very warm welcome to all of you to this very special ISCH webinar. We are in the middle of the summer. This is the 16th of July, and it is my great pleasure to welcome you all to this webinar, which comes at a pivotal moment, I think, in the evolving treatment landscape of relapsed refractory multiple myeloma. Because today's session is dedicated to antibody-drug conjugates, and this is a rapidly advancing class of therapies with the potential to reshape our therapeutic strategies. And our focus will be on belantamab mafodotin, which is a first-in-class anti-BCMA ADC that continues to generate significant clinical and scientific interest. And as you will see, and why it is special, because this webinar is inspired by two high-impact presentations delivered during the COMy 2025 Congress, which was held in May 2025, so a couple of months ago. And we will broadcast two very important lectures. First, it will be Dr. Martin Kaiser, who provided a comprehensive overview of the mechanism of action and biological rationale behind ADCs, particularly belantamab, highlighting the nuances of intracellular drug delivery and immune modulation. The second lecture will be delivered by Dr. Roman Hajek from Czech Republic, and he will present the latest clinical data with the updated efficacy and safety results. And all of these results are generating a lot of interest in this important treatment approach. But I would like to also add, as part of this introduction today, and this is a live introduction, that our discussion today and this webinar is also extremely timely, because tomorrow, July 17th, the FDA's Oncologic Drug Advisory Committee, the famous ODAC, will hold a dedicated meeting to evaluate the clinical development and benefit-risk profile of belantamab mafodotin in multiple myeloma. Additionally, you may remember that on May 22nd, 2025, the CHMP of the European Medicines Agency adopted a positive opinion recommending the marketing authorization of belantamab for the treatment of relapsed refractory multiple myeloma. These developments reflect a renewed confidence in the role of ADCs, especially when used thoughtfully in combination and with refined patient selection to offer durable responses in heavily pretreated patients. We are extremely delighted to have you with us today, although maybe many of you are already enjoying some summer holiday. And we will review really important data today about the scientific foundations and future directions of ADC-based therapy. Please feel free to engage actively with your questions and insights, and I'll do my best to answer at the end of the broadcast. So let's begin these important lectures.
I will give a background as Mohammed masterfully introduced to the mechanism of action of ADCs, these are my disclosures. And I think what I really think is worthwhile reflecting on is what a new era of trial data we have been entering with the DREAM7 and DREAM8 trials. Really trials for the very, very first time that really compared triplet against triplet combination treatments. And for the very first time, we see a fantastic improvement of progression-free survival and overall survival. So I think it is worthwhile thinking about if both of them used a triplet combination therapy, what made the drug that was changed out in this trial, so the naked antibody for the ADC in this case, what is the mechanism of action? Why is it potentially that this drug is working so much better in this combination and other combinations as well? So, antibody-drug conjugates, I think with the mechanism of action, of course, I want to give you a bit of a background to how they are really constructed. What are they? They are actually monoclonal antibodies, naked antibodies with a payload. We already had the word, and we know that at least some people in the room have an idea about what it is. So actually, as any monoclonal antibody, the antibody component of this has to be produced out of a cloned cell, of course, and then there is a payload that's actually manufactured separately, and what you then do is you bind the two together through a linker. You functionalize the antibody into an antibody-drug conjugate in linking the drug to the antibody. So we already heard about payloads, and there is actually quite a range of payloads that are available. You don't need to memorize them, you don't need to dig into this too deeply, but it's just to show, as Mohammed had already highlighted, our friends in oncology, our friends in hematology treating other diseases, of course, are using ADCs in different flavors and different versions, and actually many of them might have different payloads actually attached to them. The one that we're focusing on here is encircled with a frame there, the auristatins. That's actually the one that is now showing really fantastic activity in myeloma, but as you can see, ADCs might in the future have completely different actually applications as well, including potentially attracting immune cells, although that is not the main mechanism of action of the drug that we're seeing being active in myeloma now. So the only licensed ADC for myeloma, or soon to be licensed ADC in myeloma, is belantamab mafodotin. We have seen the trial data, some of this already, and actually the magic of this drug is what you see here. Effectively, what you have on the left-hand side is actually the original naked antibody molecule. Then there is chemistry effectively that's happening to this antibody molecule, as I just showed before, after that, with a linker that is just something that binds ultimately the payload to the antibody. And this is actually the toxin. This is the actual chemotherapy drug that is ultimately working against the myeloma here, the auristatin F. And that together leads to belantamab mafodotin as we are using it in the clinic. So what's happening and what's different with this type of antibody than with a naked antibody? Well, actually, the important bit about the antibody-drug conjugate is that it actually gets, by binding the antigen, internalized into the cell. And in the cell, then the payload gets detached. It gets effectively lysed through different mechanisms. There can be an early or a late lysis. But ultimately, what we then have is a chemotherapy drug inside your cell. And that can do its action by either disrupting the microtubules, which you are all familiar with, are important for cell separation. Any cell that tries to grow and to divide needs this. And if this is inhibited, then that doesn't work anymore. And there's also some action against DNA. And actually, some of the antibody then might get externalized again, the antigen for sure. Well, is that necessarily the only mechanism of action? It doesn't have to be the only one, although I think it is undoubtedly the most powerful and strong one that actually sends cells most into apoptosis. But then there are other effects that the antibody can have directly through its Fc region, for example. It can still activate other immune cells around it, and that can be different from ADC to ADC. But there is also an interesting mechanism that happens when you actually start killing a cell through these toxins. So here again, the antibody enters the cell and the chemotherapy drug gets released. It also causes effectively protein stress for the cell. And that leads to quite a number of surface markers being effectively as alarm signals expressed on the surface of the cell, and that can activate quite a lot of other immune cells. And this is actually quite an elegant animal model where you can actually see what you saw before, the naked antibody on the left-hand side that was ultimately leading to the manufacturing of belantamab. And actually, if you give this naked antibody, there are far fewer immune cells that are going into the bone marrow when these cells die than when you give the drug that actually has the payload attached to it. So, very powerful mechanism, different modes of action. The key one really, the payload being released into the cell in a very targeted way. And why is that so magic? Well, if we think about it, of course, drugs that are tubulin inhibitors have been around for a while. It could have been used for a while, but if we use it actually just as a drug that we give as an infusion, we have a very narrow therapeutic window or a very narrow therapeutic index. Because as we know, when this drug gets to other tissues, it can have other side effects. And this therapeutic window then gets magically wider if we actually attach it to a very specific antibody that only brings this drug to the cells that have to be targeted. And that is really the magic. You bring the payload here to the target. That brings us to the next question. What is the target? The target is the BCMA pathway, which we, of course, have heard a lot about, but I think it's so essential for myeloma cells and plasma cells that it's worthwhile always reflecting back on it, that this is really a pathway by which several growth factors can bind. There are different subtypes of this pathway of receptors that can be expressed at different stages of the plasma cell maturation, but the BCMA is the one that's most relevant for myeloma. And actually, in addition to just being used now for a drug, it actually really also serves a survival purpose for the cell. So actually, that's really clever in terms of targeting, because you don't only bring the payload to the cell, but you also inherently somewhat disrupt the survival pathway that actually that type of surface marker is mediating. Now, why is it so attractive for myeloma and an ADC, especially for an ADC? One has to say with an ADC, we want to be very, very specific that we don't bring the very relatively unspecific cytotoxic drug to other tissues. We don't want to bring them into tissues that could potentially cause other side effects. And BCMA, of course, is a perfect marker in that sense, and it is really, really highly and mostly expressed in terminally differentiated plasma cells and in myeloma cells, and particularly the myeloma cells are the ones that are very sensitive to this. Now, if we actually think about all of these features and link it a little bit with our clinical experience, and I'm probably jumping a little bit ahead to the talks that are going to be given by my very esteemed colleagues very soon, but we now have if we just think about antibodies, we have on the left-hand side a naked antibody, we have on the right-hand side a bispecific antibody, a T-cell engager, and then we have the ADC here in the middle. I think there is actually a certain sweet spot that many of us feel this drug can really serve very well, in that it has clearly a higher potency. That has been shown in DREAMM-7 and DREAMM-8, no question about that. Actually, the side effects, we will hear a lot about very specific side effects that are the corneal side effects, but actually outside of that are really also proven by DREAMM-7 and DREAMM-8, relatively limited. But then those of you who are treating people with T-cell engagers at the moment will probably have the same experience. They often have to come back really every week, especially when they start their treatment, to a clinic, because all kinds of things can happen at the start of therapy. So a colleague of mine called that hand-holding that is needed when you start these types of treatments. And I would say everyone who has used the ADC knows that that's slightly different there. Now, if we look at some toxicities, I think I'm just bringing them up here, again, not to jump ahead from my colleagues, but it is important to know, due to the mechanism of action, the very first cycle, when you get the very first drug, you bring, of course, quite a lot of this chemotherapy drug into the bone marrow, and that could be the explanation that in the first cycle, thrombocytopenia is not uncommon. Later down the line, when you have fewer myeloma cells in the bone marrow, that is less of an issue, but in the beginning at least, that could well be. There could be other theories why, but that's at least my explanation as to why thrombocytopenia needs to be watched in the first cycle. I think then something that, again, I don't want to jump too far ahead from my colleague, but the corneal side effects, I think, is very useful to see them in the context of the cornea is something like fingernails, It grows constantly back from the outside to the middle. We don't fortunately have to clip it, but it just sheds in the middle. And that whole process takes 4 to 5 weeks. Actually, if you think about it, your whole cornea gets replaced. It just takes time, and that will be something that you will hear more about. And I think as one of the last points that I want to touch on, we of course know and have heard in this meeting also a lot about CAR T cells and bispecific antibodies. And as we know, for both of these, the T cells are the essential effector that we need. And if these are exhausted, these drugs don't work that well. And this is, of course, a huge advantage if we have a drug that is using really a different mechanism of action that is not really overlapping with exhaustion of T cells, because it means that resistance to this drug does not necessarily mean that just because one targeted BCMA as well, another BCMA agent doesn't work anymore. And actually there's really reassuring data from Majestic 1, and I think there's more already with real-world data coming out as well, that we can still see very deep and also very durable remissions in patients that have had an ADC against BCMA when they're after that treated with a BCMA targeting T-cell engager. So that brings me to the summary. ADCs are clearly a very potent targeted mechanism with multiple layers of actions. Targeting BCMA with ADCs in multiple myeloma is highly efficacious. We really should not forget about what a step forward it is to have a head-to-head comparison and such a clear progression-free survival and overall survival advantage as in DREAMM-7 and DREAMM-8. The ADC payload side effects are manageable and infusion frequency dependent. Of course, you will hear much more from this about this from my colleagues. I touched on the cornea and on the thrombocytopenia, but also really important to keep in mind that they are not T-cell mediated, and sequencing is really possible. And with that, I would like to thank you for your attention.
Hello to everybody. First of all, I want thanks to organizers, especially Mohammed, inviting me. Happy to be here. It's excellent congress. You can see title of my presentation, what we have learned. This is my disclosure. And here is agenda. And I will start with just really limited introduction, because during this excellent congress, everything was reviewed. So you do agree that therapeutic landscape definitely is changing significantly. We are now moving in newly diagnosed from triplet to quadruplets even. And in first relapse, very common, starting our patients are pretreated with lenalidomide and even with anti-CD38 monoclonal antibodies. As a reflection, we have new guidelines which were several times announced here, so I do believe will be pressed soon. And if compared to last version, we have more treatment option. This is for second line. And I do believe the next version, after 4 years, will be even better, because we move with bispecific to early relapses, which is not here because the data are promising but not mature enough, so drugs are not yet approved in combinations. But simplifying things, we have like 4, I think, the strong pillars as a basis of regimen for multiple myeloma currently. Of course, proteasome inhibitors, immunomodulatory drugs, anti-CD38 monoclonal antibody, and new group BCM-targeted agents. So now the short overview, and I will be focused only on the 2 very successful randomized phase 3 trials, DREAMM-7 and DREAMM-8, which, was successful not just statistically, but the outcome is really clinically meaningful. So again, the trials were reviewed here several times, so I will be very short and quick. DREAMM-7 comparing belantamab versus daratumumab with backbone bortezomib dexamethasone, population first relapse plus, and significant better median of progression-free survival for experimental belamaf arm, close to 37 months. And as you can see, for patients refractory to lenalidomide, 25 months. So very competitive outcome. nicely separated curve for overall survival. Data are still not mature enough to reach the median, but its hazard ratio is 0.58, so it's nice. And the predicted median of overall survival is like 84 months for belamaf arm, so it's very impressive. DREAM8, belantamab, pomalidomide, dexamethasone versus bortezomib, dexamethasone, again population first relapse plus. And again, impressive data. Median follow-up is shorter than in DREAM7, so median of progression-free survival in experimental arm is not yet reached, but it was already announced here that it would be very similar as for DREAM7. So we will see on ASCO and IHA that would be data there. And again, for lenalidomide refractory patients, 24 months, I think it's very, very representative and similar result as for DREAM7. Overall survival, short follow-up, not mature enough to draw any conclusion. So what we learn? I think the learning curve was quite difficult, tough. It's required a lot of optimizations, but I think that it was hard work for GSK and for some other research group who try to optimize the regimens. So if I will go through, I first just announce the difference between DREAM7 and DREAM8 trial. Okay, so the trials looks very similar, you know, first previous treatment lines, more than one, you know, median follow-up, median progression-free survival would be similar. MRD rate lower than for bispecific and CAR-T, but significant, 39, 32%, some limited toxicity except ocular. But I want to highlight this one, the population. So if you can see, for DREAM7 exposed patients to lenalidomide, half of them, but for from DREAM8, 100%. Refractory to lenalidomide in DREAM7, 1/3 of patients, in DREAM8, 81%, so it's significant different. And if you will be focused on anti-CD38, just limited for DREAM7, in DREAM8 exposed 1/4 of patients, most of them refractory to anti-CD38. Slightly different dosing. So what we learned? We learned that infection is not a big issue if you are using belantamab. You know, the infection rate is high in any trials in myeloma, because the treatment. But if you compare the control arm with belamaf arm, it's very similar for any grade. 70% versus 67 for DREAM7, 82 versus 68 for DREAM8. Discontinuation of study treatment, 10% versus 7% in DREAM7, and similar low data experimental and control arm for DREAM8. Grade 3 was significantly higher in belantamab arm, but was manageable. So in some analysis, we know from analysis that it was used low rates of immunoglobulin substitution through both trials compared with very intensive supportive care required for BCMA targeting bispecific antibody or CAR-T therapy, which is mandatory currently. Whilst grade more than 3+ infections were more common with belamaf containing regimen, infection had low fatality rates and high rates of resolution. And there were low rate of opportunistic infection across all treatment arms. So generally, these results suggest that belamaf containing regimen have a tolerable infection profile compared with other bispecific or CAR-T therapy targeting BCMA, which is, I think, really important for, you know, routine use of the drug. What about administration frequency? I will not talk about toxicity, but I think it's really critical to educate physicians how to use belantamab in real life, because we all do believe that the belantamab will be soon approved and reimbursed and used. And especially not hematologists are quite expert, but in many countries, oncologists will be used belantamab. And it's critical to educate them and increase the confidence that the critical step is to prolong interval between the dosing of belantamab, reducing significantly ocular toxicity without losing efficacy. And this, I think, critical point for practical use of belantamab. We have data, more data coming, so GCK doing a really excellent job in this field. This is data from DREAM7, impact of dose modification, and I just want to highlight it that median weeks between doses increase with treatment duration, as you can see here. Starting point is as suggested 3 weeks, but then increasing to 6, 8, up to 12 weeks. Dose delay did not have a negative impact on progression-free survival. If I will talk about DREAM8 study, I can just repeat what I said. Again, original starting dosing frequency 4 weeks, increasing from 8 up to 12 weeks, so 3 months. And again, dose delay did not have an impact of progression-free survival. And if you calculate the number of administration of belantamab during 2 years and compare this frequency with other strong regimen currently used, that's, there's some advantage from patient's point of view, because the number of belantamab administration in DREAM7 is 30 in reality, in DREAM8 16, and if you compare it with other regimen, it's significantly lower numbers. So otherwise, it can potentially increase quality of life of our patients. So ways position of belantamab regimens, and of course we will be now focused on patients to refractory to lenalidomide. Currently, probably the best what we can use is anti-CD38 plus carfilzomib and dexamethasone, and if you are lucky and cilta-cel is available in your country, that's the clear option. But now it's coming belantamab, and if you compare all other regimens available, then I just highlighted what is, I think, clinically meaningful, because all other regimens achieving, I think, suboptimal results, usually 1 year of median of progression-free survival plus. So clearly, anti-CD38 carfilzomib dexamethasone, belantamab-based regimen, and cilta-cel will be treatment options. Of course, we have to wait on approval of belantamab. So my conclusion is very simply, belantamab combination with VD or pomalidomide dexamethasone show excellent outcome in early relapse multiple myeloma compared to control arms. And these are regimen, one of the most effective treatment choice in patients refractory to lenalidomide. Belantamab containing regimen have a tolerable infection profile compared with other BCMA targeting therapies, and with no mandatory substitution of immunoglobulins, and limited frequency of belantamab administration in real world make belantamab regimen convenient for patients, and I have to say also for staff. So I stop here and thank you for attention.
Well, thank you, Martin, and thank you, Roman. This was, well, we deliberately wanted to make it short because we know that it's summertime and we wanted to have just a very straightforward, to the point webinar. Still, I think we can take a few questions. And one important question here I can see from Dr. Lucian Welter, whether the use of belantamab can affect the later efficacy of CAR T cells. Well, obviously, we are lacking these controlled, I would say, prospective comparisons with controlled. And this is a question about the optimal sequence. However, I think, as highlighted by Dr. Kaiser, given the mechanism of action, if BCMA is expressed, in the case of belantamab and antibody-drug conjugates, you're using the receptor, the target, to allow the entry of the drug, and then you have the chemotherapy-like effect. When it comes to the cellular immune therapy, I think the process is relying on the BCMA signaling pathway, where actually you're trying to alter the role of BCMA in the survival of the malignant plasma cell. So to make a long story short, I don't believe there's going to be concerns between the use of CAR T cells or the use of belantamab. Also, we need to acknowledge, and here we have another question from the audience about how to choose between the bispecific, the CAR T cells, and the antibody-drug conjugates if all of them are targeting BCMA. And this is, of course, a $1 billion question, but I think it's not going to be as complicated as it appears. Well, in general, people and the experts, whenever possible, are recommending to use CAR T cells because it's a single-shot treatment. However, we need to acknowledge that CAR T cells are not available everywhere. Not all myeloma patients can receive CAR T cells. One simple example is the ability to administer, for instance, fludarabine in those patients who have severe kidney dysfunction. And we know that there can be some contraindication there. Also, there is a need for hospitalization because obviously, at least in European countries, CAR T cells are not made on an outpatient basis. Also, the very old patient are not receiving, in general, CAR T cell. So this is why I believe many patients it's not an opposition between CAR T cells, bispecific, and antibody-drug conjugate. I think the results of the DREAM7 and DREAM8 trial, as shown by Dr. Hajek, are very convincing when it comes to giving into early relapse the belantamab-based combinations. I can see here in the chat another question about whether you would rather prefer an anti-CD38 based combination like carfilzomib plus isatuximab or daratumumab at time of relapse, or belantamab based combination, and assuming CAR T cells are not available. Well, obviously, this is an important question. And again, we do not have any head-to-head comparison between these different combinations. So we need to acknowledge that the choice is going to be maybe empirical, but also we need to acknowledge that, for instance, older multiple myeloma patients who are receiving, for instance, daratumumab lendex, the Maya trial, or receiving a quadruplet like isatuximab VRD, like in the IMROSE trial, or daratumumab VRD, bortezomib lendex plus an anti-CD38, like in the Cepheus trial. Well, at some point, these patients are going to be refractory to an anti-CD38 monoclonal antibody. And this is where targeting BCMA is actually more attractive and is going to be the optimal choice. Another feature that was highlighted, and again mentioning because we have a question about this, whether in the older population there is a benefit for the longer cycle, the extended cycles, and this is a true benefit with belantamab because actually you can end up having 4 maybe or 5 administrations per year. Another question here from Dr. Jim O'Mell, to whom I say a big hello. Jim, wouldn't belantamab be a great bridging drug for patient waiting for CAR T production? Short answer is yes, indeed, absolutely, and this need to be tested. And I think when the drug will become available and approved, it is already on the right path in Europe, and tomorrow we'll have the ODAC meeting in the US. So I'm quite optimistic about the complementarity, the possible use of all of these options. We have here a question about from Dr. Iqbal about ocular toxicity. Well, to be honest, historically, ocular toxicity has been a major limitation of the use of belantamab. However, today, as you have seen, with some precautions, especially the the longer cycles, actually, this is not anymore, I would say, a matter of concern. We will have the opportunity as part of the ICH activities and the Multiple Myeloma Academy by the ICH to dig a little bit deeper into the role and how to prevent, how to minimize the risk of ocular toxicity. But definitely, I think compared, if you're referring, Dr. Iqbal, to something from 4 or 5 years ago, this is not the case anymore today based on the DREAM7 and DREAM8 trial. We have another question here about cytotoxicity and thrombocytopenia and leukopenia. Well, obviously, like with any anti-myeloma therapy, we may experience cytopenia, especially in the relapse setting. And there may be many reasons for this. Either, of course, the drug you are using, but it could be also related to the bone marrow reserve, which we know is going to be poorer at time of relapse. But again, this can be managed, I would say, relatively easily by hematologists. Some patient may require a little bit of GCSF. That's not common or systematic to everybody. In terms of thrombocytopenia, in general, we rarely need any transfusions. It's more about the use of TPO agonist. Well, there are still many, many questions, but we'll have other activities, I think, dedicated to antibody-drug conjugate. For the sake of time, I would like to conclude and extend my sincere thanks to all of you for your engagement, and of course, to the beautiful talks from Dr. Kaiser and Dr. Hajek. Very clear, very insightful presentations. And we have explored today not only the mechanistic elegance of antibody-drug conjugate, as so expertly detailed by Dr. Kaiser, but also, I think, the clinical promise of belantamab mafodotin, as presented by Dr. Hajek, reminding us that innovation in multiple myeloma is thriving across both the bench and the bedside. And as I mentioned in the introduction, this discussion comes at a particularly meaningful time with the FDA ODAC meeting scheduled for tomorrow, July 17th, and of course, the recent positive opinion from the CHMP and EMA approval. So in Europe. So it's clear that antibody drug conjugates are once again at the forefront of therapeutic innovation, offering renewed hope for patients with relapse refractory disease. Moving forward, I believe the key to unlocking their full potential will lie in optimal patient selection, rational combination strategies, but also real-world evidence to guide the clinical use beyond trial setting, as reflected by many of your questions. And I think I remember we have the Scientific Steering Committee of the ICH is planning something about the importance of real-world evidence, probably for the months of September or October. At ICH, we remain committed to providing a platform for these timely conversations, bridging the gap between discovery and clinical practice. Thank you for joining us, and we look forward to seeing you at our upcoming events. Stay safe, stay inspired, and keep pushing the boundaries of what is possible to all patients everywhere. Thank you very much.